Good evening, everyone. Can somebody please tell me if I'm audible and also you can see me? Can you people? Is my video? Can you see this, or is there any problem with the video or the audio?
No, ma'am.
Okay, thank you. We'll just wait for a few more minutes to let others join, and then we can start. So, yeah, I think we can start. Good evening, everyone. I am Nisha Barke from IISc. I'm going to be your TA for this NPTEL online course Drug Delivery Principles and Engineering, which I think you must be knowing is hosted by Professor Rajat Agarwal in Department of Bioengineering in IISc. I'm actually his PhD student. So what we are going to do in these sessions is we are going to discuss some of the points or some of the aspects which have been covered in the lectures which you guys must be following every week, and at the end of this discussion we'll also try solving some of the problems that can come up with this drug delivery course. So these sessions will help you. So I believe most of you would be taking an exam at the end of the course. So these sessions will help you first of all clear your concepts about drug delivery, know more about this field, and it will also help you in solving some of these problems which can ultimately help you with the final exam. So does anybody have a doubt before we start, or we can directly start with the session? You people can stop me at any point, just unmute yourselves and let me know if there's a question, and we can then continue with the discussion. If there are no more doubts, I'll just share my screen and we can start with this particular session. So I'm going to host a session every Tuesday from 6 to 8 p.m. Parallelly, there will be a session on Monday as well from other TAs. So in case if you miss one of my sessions, you can follow it up with their sessions as well. Also, I'll be uploading the presentation as well as so yeah, for your kind information, this video is being recorded because I'll be uploading it on the YouTube channel as well. So in case some of you miss any of these sessions, you can then follow it up later on. But yeah, in these live sessions you can just try asking your doubts, or we can have a discussion about the topics. So yeah, I'll just share my screen and you people can let me know if you can see the presentation. Can you see this in a slideshow mode? Somebody can just let me know. It's visible, right? Guys, anyone, just so that I don't. Okay, I think you people are replying. Okay, yeah, sure. Yeah, thanks for letting me know. So we can just start with this. Let's start with some of the key points that are covered in the week 1 lectures. So you must have gone through all these lectures as well as the notes. This is just going to be a recap which will help us solve the problems at the end of the session. So first of all, let's start with the field of drug delivery and why we need to study this. Drug delivery is nothing but the appropriate administration of drugs for improving patient health. And so the question comes to our mind of why, first of all, we are studying drug delivery, what makes it more important. So as you must be knowing, there are various diseases which humans are prone to get, and we also have treatments available for them. Some of these diseases, most of these diseases are curable as well as preventable. Then why do we need to study drug delivery? So despite of having current treatments taking very well care of these diseases, we see that some of these treatments have the drugs used in these treatments have severe side effects. Some of these treatments are for a very long duration, and also frequent administration of drugs is seen in these treatments. For example, I'm sorry for this constant changing of the screen. I need to admit new people as and when they come up. So yeah, we were talking about these current treatments. So yeah, there are certain disadvantages with the current treatment. For example, let's take TB treatment, tuberculosis treatment. That's what I work in. So TB is curable as well as preventable, but the treatment follows for 4 to 6 months, and people have to take medicines every day for these 4 to 6 months. And it sometimes happens that after 2 to 3 months, because of the side effects of these drugs, people will start ignoring the drug dosages. They will be like, no, now I feel better, now I don't need to take these drugs. And then this leads to complexities like it can lead to resistance of the bacteria to these particular drugs, and so forth. So to avoid all of this, that's where drug delivery comes into picture. What our goal here is, we have to optimize the administration of these drugs to remove some of these disadvantages. For example, you design a drug delivery technique for TB in such a way that when you can give only one dose and it should be enough for a month. So instead of taking medicines every day, the patients can just take that one dose which will last for a month, and they can only take 4 doses and the treatment will be completed. So that's the goal of drug delivery. So to understand this field as well as to come up with new techniques to deliver drugs, we need to first go through some basic concepts. So some of these are I've listed out here. So First is pharmacokinetics. So what does pharmacokinetics mean? It means how your body is going to react to a particular drug. So now whatever drugs you have, most of these will be a foreign material for the body. You're injecting it or ingesting it into the body, so the body is definitely going to have some reaction to this drug. So how do we study that? We study that, so for example, your body is first of all going to absorb some of this drug. It will distribute it all over the body, because now if the drug enters your bloodstream, blood will flow to each and every part of your body, and then that's where the distribution of the drug comes into picture. Also, some of this drug, being a foreign material, is going to be metabolized by your liver. So your body will try breaking it down into small particles, and it will try eliminating it out of the body, because your body thinks this is a foreign material. So all of these aspects come into pharmacokinetics. Why this becomes important is because until and unless you know how the drug is going to be distributed in the body, how it's going to be eliminated from the body, you will not be able to design what dose you should be giving, at what frequency you should be giving this dose, how longer the treatment should follow. So all of these aspects we need to study to understand pharmacokinetics of a drug, and then that will help us in designing a very good drug delivery system for a particular application. So now there are various factors or various parameters of a drug that affect pharmacokinetics. Some of these are solubility. Okay. Does anybody have a question? Yeah, so some of these factors are solubility, how soluble your drug is in water, because once it enters the bloodstream, the drug has to have good solubility in water, because most of our body is made up of water. That's how your drug is going to be distributed to each and every part of the body. So you need to know what is the solubility of the drug, if it's a hydrophilic drug or a hydrophobic drug. Let's say you're trying to design a drug for a particular treatment, and you have designed a very good drug, but you come to know that it is very hydrophobic in nature, which means it is not very, doesn't have very good compatibility with water. And you see that once you administer the drug, it gets precipitated out. So no matter how effective your drug is, until unless it is soluble, well enough soluble in water, it is not going to help you for the treatment. So all of these factors of the drug, the parameters of the drug, will affect your pharmacokinetics. So there are a few other concepts for studying drug delivery. First is first pass metabolism. So what does first pass metabolism mean is, this is in context of drugs that are given orally. So what happens when you take a drug orally? Most of it, as it's going through the digestive tract, most of the drug gets delivered to the liver via a particular vein, and and that's where it gets metabolized. So most of this drug is already lost when you are giving drugs orally. So this effect is known as first pass metabolism. I'm sorry to take up this question. I hope you people don't have any objection with this. Somebody is asking, there were 2 sessions mentioned in email from 6 to 8 p.m. and 7 to 9. So attending any one of these sessions is okay. Yeah, so you can attend any of these sessions depending upon whichever time is comfortable for you people. So if you guys want to attend, so this session will not go till 8 p.m. I believe. Yeah, I think we can take this session from 7 to 7:30. It's okay. And if you guys still want to join the other session, that is also perfectly fine. Since this is the first session, I suggest you attend both these sessions and see whichever time you are comfortable with. So every Tuesday you are going to have 2 sessions. One is 6 to 8, one is 7 to 9. I hope that clears it. Yeah, so we were talking about first pass metabolism, which we just discussed, and then there's another concept known as bioavailability. What this means is, bioavailability essentially tells you how much amount of drug that you have administered will actually reach the site of action. So for example, let's say you're treating TB. TB is essentially a disease that affects the lungs primarily. So let's say I'm taking a drug orally. So I have taken my medicine. Now I want the drug to reach my lungs. Not 100% of the drug that I have taken orally is going to go to the lungs and then kill the bacteria there. So some amount of drug will definitely be lost in this route of administration. So this is where bioavailability comes into play. It tells you how much amount of drug will actually reach the site of action. We will discuss about these concepts in detail as well when we are trying to solve some of the problems from the assignment. Then there is something known as elimination. So elimination is nothing but your body trying to eliminate the drug. So the rate of elimination could follow a zero order or a first order reaction. Zero order meaning the rate of elimination does not depend upon how much amount of drug is there in the body. It's a constant rate of elimination. First order is uh, the rate of elimination will depend upon how much amount of drug is already present in the body. Then we have a concept of half-life. Half-life is nothing but the time it takes for the body to eliminate half of the amount of drug that you have administered. And then clearance is nothing but there is a particular process where your body will try clearing the drug from all the tissues that it has already reached. Yeah. Then there are few parameters to characterize a drug. These become important when you are, let's say, trying to discover a new drug, or you also need to know these when you're trying to come up with a new drug delivery technique. So these are ED50. ED50 is nothing but effective median dose. So what this dose tells us, this is the amount of drug in when given 50% of the population that you have given this drug to shows the actual effect, shows the therapeutic effect of the drug. Then we have TD50. This is the amount of the drug which when given to a population, 50% of that population will show toxic effects for the drug. And then there is LD50, which is the amount of the drug when you give it to a population, 50% of this population is going to die because of this amount of drug. So all of these help us in coming up with a value known as therapeutic index. So this is how you characterize a drug. You need to know the therapeutic index of your drug. What is therapeutic index? It is nothing but the ratio of either TD50 or LD50 value to your ED50 value. So you need to have a very high amount of therapeutic index for the drug to be actually effective. So what does this mean? A high value of therapeutic index tells you that your drug is going to be toxic at a very, very high dose, but it is also going to be very potent, very effective at a lower concentration. So that's what you should be looking for when you're trying to come up with a new drug. Then we move into so these are some of the basic concepts, and then we start discussing about the actual drug delivery systems. One of it is prodrugs. One of it is prodrugs. So we'll also talk about products in one of the questions in the coming assignment. So products are compounds which get metabolized into the active drug. Why do we need this concept? So it can be used to improve bioavailability, or you can use prodrugs to target a particular site. So as I was saying, for example, let's say you discover a very good drug and you come to know that it's a little bit hydrophobic, not all of it is becoming bioavailable at the site of action. What you do is you conjugate a hydrophilic molecule to this prodrug, then you deliver this product. So now it has become a little bit more hydrophilic, so the solubility of the drug increases, and then more amount of drug will reach the site of action. This is one example of improving bioavailability using prodrug concept. Then you have a concept known as controlled release. So what does controlled release mean? It means that when you are giving drug to a patient, you don't want all of that drug to be available at one particular time. You want to have a certain amount of control over how much amount of drug you want to be released in the body at a particular time. So this helps us in having a very sustained release. For example, as I was talking in case of TB, let's say I have a formulation, I have taken my drug. I don't want all of it to be bioavailable at once. I want the drug to be released in my body over a week. So that will give me a certain amount of control, and it will help me in reducing the dosage frequency. So now instead of taking drug every day, I have to only take it once a week. So this is where controlled release comes into play, and we can achieve this using biomaterials. So so biomaterials are so some of these polymers are also termed as biomaterials. So what are polymers? Polymers are nothing but it's a long chain of repeating units which we call as monomers. Sorry to take this question. Somebody is asking, will I share the notes or do you have to note them? No, you don't have to note anything. You just keep on listening to this lecture. I will be sharing the PPT as well as a recording of this particular video, so you can go back and check it anytime you want. Right now you don't have to note anything. I hope that's clear. So we were talking about polymers. So polymers could be synthetic or natural. For example, your protein is a polymer. So protein is made up of repeating units of amino acids. That's one example of polymer. So these polymers help us in designing a drug delivery method which will help us achieve controlled release for a particular application. So somebody trying to say something? So yeah, so there are various ways of classifying polymers. This classification can be based on a chemical structure or on its properties. It could be based on different basic properties of polymers. So for example, depending upon what your polymer is made up of, let's say you have only one particular monomer that is always repeated in a polymer, that could be one type. Then this is known as a homopolymer. Then you can have two different monomers randomly arranged in a polymer, that is known as random copolymers. So in this way you can just classify different types of polymers. That helps us study them better. And then there are basic properties of polymers such as molecular weight, its crystallinity, its melting temperature, whether your polymer is going to be degradable or not. So all of these properties, basically, why do we need to study them? They kind of help us in selecting a particular polymer. So there are a number of polymers that are available for me. I don't know which one to choose for which of my applications. So when I study all of these polymers, I'll study all of these parameters, these properties of a particular polymer. That will help me choose one or two polymers for the optimum drug delivery technique that I am trying to look for. So you people must have already gone through some of these concepts. This might be a repetition for few of you, but I think that's enough of discussion, and we can move on towards some of the questions in assignments. Is that okay with everyone? Does anybody have doubt before I move on towards the questions? So the questions we are going to discuss now are not from your current assignments. Of course I can't do that. I think you have two assignment deadlines coming up, which you have to submit on 8th August, I guess. So these are some of the questions from previous year assignments that we'll be discussing in these sessions. Shall I go ahead with the questions, or does anybody have a doubt before I move on? No? Okay, I'll try taking up these questions one by one. Let me know if you can see the question. I'll also read it out for you. And after each question, I'll take a pause to let you people answer it. If anybody wants to answer, you can just try unmuting yourself and try answering it. If not, of course, we'll be going to discuss it. So yeah, the first question is, a prodrug can be, and we have 5 different options. You have to choose 2 options. It can be a prophylactic drug, or it can be a drug conjugated to enzymes cleavable by biological systems. It is a prototype member of a class of drugs. It is the oldest member of a class of drugs. It's an inactive drug that is transformed in the body to an active metabolite. So there are 2 correct statements here describing what a prodrug is. I'll let you people take some time off, read it, and if anybody wants to answer, you can let me know. Someone trying to answer? I'm sorry, I can't see your names. I'll have to keep on flipping in the windows. Yeah. Anybody is trying to answer? Are you people answering in the chats? Okay. Yeah, someone is saying 2 and 5. Someone is saying drug conjugated to enzyme. Okay. I think 2 and 5 is what somebody is answering. Someone has answered option 2. I think someone else is trying to answer. Can you be a little bit louder? I can't see if you have raised your hands because I need to keep on flipping through the windows. So it's okay. You can just unmute yourself and try speaking. Or you can type in the chat box. I won't be able to see your raised hands. Yeah, Pavan, you can unmute and tell me your answer. Sorry. Yeah, this is the question. Okay, any more responses? Thank you to everybody who's trying to answer. Yeah, some of you are saying 2, 5 and 2. So make sure whenever you'll be reading such questions in exams, it says you have to give 2 correct options. So you have given 1 option, you will also have to select another. Okay, someone is saying 5, 2, 5. Okay. Thank you all for answering. I'll just display the correct answer. Most of your, yeah, I think all of you are correct in giving option 2 and 5. So second option, so we'll just discuss what prodrugs are in a bit more detail. Right, so products are nothing but compounds that get metabolized into active drugs. So let's say this is your active drug and you conjugate it with some molecule. So this entire thing will be known as prodrug. And what happens is once you deliver this prodrug in the body, some enzymes which are capable of cleaving of this particular bond, they will act on this, they will cleave this bond, and that will release your active drug. So this may result in increasing bioavailability, like we were discussing. Let's say your drug is very hydrophobic, you want to make it little bit more soluble. So what you do is you conjugate it with hydrophilic molecules. So that will increase its solubility, ultimately increasing its bioavailability. It also helps you in doing a targeted drug delivery. Let's say, for example, you have made a prodrug, you have conjugated it with some ester bond here, and you know that enzymes esterases would be present in, let's say, one particular, any other enzyme, esterases or such enzymes would be present in only one specific part of the body. So once you give this prodrug, once you deliver this prodrug in the body, it will release active drug only in the presence of these enzymes. So it has to reach that site first, and that's where the active drug will be released. So this helps us in minimizing some of the non-specific side effects that the drug might have in some other parts of the body. That's where your prodrugs actually help you. So let's look at the answers. So the correct answer for a prodrug, it's a drug conjugated to enzymes cleavable by biological systems. I think there's some typo here. It's a drug conjugated to molecules. It doesn't necessarily have to be conjugated to an enzyme. It can be conjugated to any other protein, it can be conjugated to any polymer, and that will be cleavable by the biological systems. And of course, an inactive drug that is transformed in the body to an active metabolite is known as a prodrug. Is this clear? Is this explanation enough for you people? Or like, should I go over it again?
Ma'am,
Yes, somebody is trying to say something.
I have a question.
Hmm?
What is the meaning of prophylactic drug?
Okay, prophylactic drug is nothing. So first of all, prophylactic treatment is something which you give before a person gets the disease. So it's like a preventive therapy. So for example, let's take TB treatment. Let's say I am a TB patient. So the doctors will prefer my family members whom I live with that they should take a prophylactic treatment. So this is done to avoid my family members getting TB from me. So that's a prophylactic treatment. The drugs that you use for prophylactic treatment are termed as prophylactic drugs. Clear? Is this okay?
Sure, ma'am. Yeah.
Yeah.
Thank you, ma'am.
Thank you. If there are no more doubts in this particular question, I'll move on towards the next one. If anybody has doubts in this question and we have already moved on, it's okay. At the end of the assignment, we can go over the questions once more if somebody has missed some of these questions. Yeah, so second question is, which of the following drugs are absorbed in the greatest amount? Again, we have 8 different options here and we have to choose two options from these. Let me know if you have the answer. I'll stop for a while. Anyone who likes to answer can write it in the chat box, or you can let me know. Okay, don't raise your hands, just unmute yourselves and let me know the answer. Srinidhi is saying 3 and 5. 3 and 5. Okay, we'll wait for others also to answer, and then we can discuss the correct option. Thank you for answering.
3 and 5, ma'am.
3 and 5. Okay. Any other people have any other options other than 3 and 5? Okay, bunch of you have answered after Srinidhi. Okay, most of you are saying it's 3 and 5. Somebody is saying 2 and 5. Somebody is saying 8. Okay, what is 8? Okay, cool. So let's look at the answers. So the correct answers, the correct options are 3 and 5. So whoever has answered 3 and 5, absolutely correct. We'll look at these options in a bit more detail. So as I was discussing, what exactly is bioavailability? It tells you how much amount of your drug will actually reach the site of action, which will give its therapeutic effect. So one rule with bioavailability is, if you are giving any drug intravenously, we yeah, so if you're giving any drug intravenously, 100% of that drug will be bioavailable, because wherever the drug has to reach, it has to reach through your blood circulation. And whenever you're giving a drug through IV route, everything that is there in your injection goes into the bloodstream. So we consider 100% bioavailability for any drug that is administered through IV route. And then we determine the bioavailability of a drug for different routes based on this formula. So it is nothing, it's defined as how much amount of drug will be the AUC of the drug through that particular route compared to the AUC given by IV route. So this formula then helps us in answering this question. So the first option is 100 mg of drug is administered and the bioavailability is 0.7. So what will I, according to this formula, what will I say? So my bioavailability is 0.7. I need to find how much amount, what is the AUC with the route. I already know 100% of the drug would be bioavailable when given intravenously. So I'll consider 0.7 is equal to x upon 100. So that gives me 70. So that means 70 mg of the drug will be absorbed by this particular route. Similarly, if you keep on doing this for the first four options, let's say 150 mg of the drug is administered and bioavailability is 0.5, you just multiply it, it will give you 75. So 75 mg of the drug would be absorbed. Then for 200 mg drug that is administered with a bioavailability of 0.4, you multiply this, you'll get 80 mg. And yeah, so for the fourth option would be, you've given 250 mg drug, bioavailability is 0.2, and it will give you 50 mg of the drug that is being absorbed. So from the first four options, if you will see, only the third option gives the highest amount of drug that is being absorbed, that is 80 mg. So we have one answer with the first four options, that's option number 3. Now let's look at the rest of the four options. 80 mg drug is administered by IV, 80 mg is given orally, 20 mg is given orally, and 20 mg is given through intravenous. So we already know that whatever is given intravenously, 100% of it will be available. So that means 80 mg of the drug given intravenously, all of that 80 mg would be absorbed in the body. So then from the rest of the four options, we know that this is going to be the maximum absorbed drug. So then we have our two correct options. One is 200 mg drug administered with a bioavailability of 0.4, and then 80 mg drug administered intravenously. Is this clear? I hope it's clear. If not, then I can go over it again. So this particular question then tells you that so if you see in between options 3 and 4, you'll see that some might argue that fourth is the correct option because we are giving more amount of drug. No matter if you are giving more amount of drug, if the bioavailability is less compared to this, it will always be that the drug absorbed in this particular example will be lesser than the third option. Is this clear? I hope I'm making sense and you guys are understanding. Thank you for answering in the chat box. Any doubts with this? Or else I'll move on to the third question. Okay, cool. The next question is, which of the following equations depict a second order reaction? Again, we have to choose two options. Let me know if anybody knows the answer. Anyone? Oh, you've started answering, is it? Okay. Most of you are saying option B. Some of you are saying 2 and 3. Some of you are saying 2 and 4. What is 2? What is 4? Okay. Okay. Anyone else? Alright, most of you are sure that it's option 2. The confusion, I think, is between option 3 and 4. Okay, so let's look at the answers. The correct answers are option 2 and 3. So whoever has answered 2 and 3, correct. So let's discuss this. So what exactly does an order of reaction mean? It just, so this is a particular rate equation. That's how you write a rate equation. And so the order of any reaction tells you how the rate of reaction is going to be governed based on the concentration of the components, concentration of your compounds. So let's take the first options. Here it says the rate would be constant. It means it's a zero order reaction. The rate does not depend on the concentration of any compound that takes part in this reaction. So it will have a constant rate of equation. Definitely this is not a second order reaction. Let's look at the option 2. It's given as R equal to K. K is nothing but your rate constant, and we have two species here, C1 and C2. Now the way you calculate order of reaction is you just sum up the exponents of these two concentrations. So here it will be 1, here it will be 1. You take a sum of it, it gives you 2. So this is definitely a second order reaction. What this tells us is that the rate of this particular reaction will be governed based on the concentration of both these species.
That's it.
Sorry? Okay. Then so we have one correct answer here, option 2. Then option C is the rate of reaction is K, again rate constant, C1, C2 raised to 0.5, C3 raised to 0.5. Again, to calculate the order of this reaction, we have to take a sum of all the exponents. So here if you take a sum of 1, 0.5 and 0.5, it will again give you 2. So this is also a second order reaction. So we have our correct answer here, option 2 and 3. If you look at option 4, this is R equal to K and C1. The exponent of C1 will be 1. So this is essentially a first order reaction. And then you have the last option, R equal to K, C1 raised to 4. This is not a second order reaction. So the correct answer is option 2 and 3. Is this clear? Okay, somebody has a doubt regarding previous question. Just hold on to that. We'll just finish all the questions first, and then we can revisit the second question, and we can discuss in detail there. Is it okay, Murari? I hope I'm pronouncing your name correct. Yeah, we'll come back to the question. Let's first finish off the questions in this assignment so that it's not a repetition for others. Right, so if there are no doubts in this question, I'll just move on. Is it okay? Okay, I'll take that as a yes. Okay, the fourth question is, which one of the following will have ease of permeating through cellular membranes? Will it be a drug with higher partition coefficient and is highly polar, drug with higher partition coefficient and is not polar, drug with lower partition coefficient and is not polar, drug with lower partition coefficient and is highly polar, hydrophilic drugs, highly charged drugs, or polar molecules? So we have to choose one correct option here. Which of the following will have ease of permeating through the cellular membranes? Let me know if you people have the answer. I'll wait for a few minutes.
It's
It's option 2, ma'am.
Option 2, drug with high partition coefficient and is not polar. Okay, we have one response, that is option 2. Anybody else would like to try? Thank you for answering. Okay, most of you are trying to answer as option 2. Somebody has option 1. Okay, we'll discuss. People still answering? Okay, I'll discuss this. So yeah, the correct answer is option 2, drug with higher partition coefficient and is not polar. Yeah, let's look at the concept of partition coefficient. So partition coefficient is nothing but a way to characterize a drug. So it is the ratio of concentration of a solute into immiscible or slightly miscible liquids when it is in equilibrium across the interface between them. So how do you calculate a partition coefficient? So you take the drug, you try dissolving it in a hydrophobic phase, which we call as an oil phase, and you also dissolve this drug in water phase, and you see how the drug partitions. If the drug is very hydrophobic, it will try being with the oil phase because it doesn't like water. If the drug is hydrophilic, most of the drug would be in the water phase. So partition coefficient is nothing but it tells you how the drug partitions based on its environment. If it's hydrophobic, it will go more towards the oil phase. If it's hydrophilic, it will stay in the water phase. So this particular concept of partition coefficient helps you in characterizing a drug. It tells you what would happen to the drug and how it will react to which part of the body. So let's look at the question once more. It's trying to ask us which of these following drugs will be for which of these following drugs it will be easier to permeate through the cellular membrane. Now you people must be knowing the architecture of cellular membrane. It is mostly hydrophobic in nature. So to pass through this hydrophobic cellular membrane, the drug has to have higher partition coefficient, right? So more the partition coefficient, more is the drug hydrophobic. And related to polarity, so this is the concept for partition coefficient. With respect to polarity, we know that if the drug is less polar, it will suppress the ionization. The drug will not be able to ionize, and this will increase the lipid water partition coefficient. So for your drug to easily pass through the cellular membrane, it has to have higher partition coefficient, and it shouldn't be polar, so that it doesn't ionize. So this then helps you in designing drugs better. So if you have a hydrophobic drug, you know that most of the drug will pass through the cellular membrane. It will be uptaken by the cells. This becomes important in few of the drug delivery applications. Is it clear? Okay, I don't see any doubts in the chat box. Thank you for answering the questions. Is the concept of partition coefficient clear? If anybody has doubts, I can stop at this question, and for this particular question we can discuss again and then we can move on. Okay, there seem to be no doubts here. I'll move on towards the next question. Next question is, bioavailability by definition is defined as, I think we just discussed this concept in the previous question, so most of you should be able to answer this. Again, choose only one option. Let me know in the chat box, or you can unmute and let me know the answer. Okay, people have started answering for this question. A lot of you, I think most of you have given option 1. What is option 1? AUC of the route divided by AUC of IV. Yeah, so we'll not spend much time here. We have already seen how bioavailability is defined. It's defined with the AUC of the route, whichever route you're trying to deliver the drug through, and AUC by IV. Because 100% of the drug becomes bioavailable, so we always compare bioavailability with respect to IV route. So whoever has answered first, absolutely correct. Is anybody trying to ask a question? Any comments? Okay, nothing much to discuss here. We'll move on towards the next question. Next is VD of a drug is known to be 30 liters. How the drug is expected to distribute in the body? So if you people have read about volume of distribution, you should be able to answer this. Again, we have to choose only one option from this. So if you have read through the question, please let me know the answer.
It's option.
It's option, sorry, you were not clear. Can you please repeat the answer?
Option 2, ma'am.
Option 2. Okay, in total body water. Thank you for answering. Does anybody have any other answer other than option 2? Okay, most of you are answering option 2. Thank you for answering in the chat box. Anyone else thinks it's other than option 2? I think not. Okay, yeah, you all of you are correct. The correct answer is in total body water. So what exactly is volume of distribution? It tells you where exactly is your drug going to stay in the body. So it assumes that the entire patient body is one single compartment, and depending upon the parameters, the properties of the drug, it will then try going to different parts. For example, let's say there is a drug called warfarin. I've just taken this from the lecture slides from the professor itself, so this might be a repetition for some of you. So we know that volume of distribution for this particular drug is 8 liter, and we know that this is roughly the same amount of blood that is present in a human body. So we conclude that most of the drug, it has a very high plasma protein binding capacity, so most of this drug will be in the bloodstream itself. In contrast to that, let's say there is another drug called chloroquine, and it has a very high VD of 15,000 liter. What does this mean? It means that most of this drug will be in the total body fat, and it's a lipophilic molecule. And then we have another example. For example, let's take ethanol, and it has a VD of 30 liters. So it tells us that most of this the ethanol will be present in the total body water. So that's what the question also is. So the drug is known to have a volume of distribution of 30 liters, so we can make an estimate of, because 30 liters is roughly this amount of total body water in a human body, so we make an estimate that it is expected to be in the total body water in the body. Is this clear? Any doubts here? I think not. Then I'll move on towards the next question. The next is for a given drug, fractional rate constant is given as 0.1 per hour. The apparent volume of distribution is 10 liters and clearance is given as 1 liter per hour. We have to find out the half-life of the drug. This is a numerical sort of question. So if you people know the formula of how volume of distribution is related to clearance and half-life, I think it should be easy to solve. Give it a try. If not, we'll discuss. I'll keep the screen as it is for a few seconds for you to note down the values. Anybody would like to tell me the answer? Are you people typing in the chat box? Okay, yeah. Oh, you guys have already started answering. Okay, option 2. Some of you are saying option 2. Thank you for answering. Anybody else has solved it and would like to answer? Any other answer than option 2? I don't think so. I think most of you have figured it out. I'll still wait for a few more seconds. Is anybody trying to answer? Okay, I see that most of you have answered correctly. The correct answer is 6.93 hours. So the way to go about this question is, I've just written it down for you. So we first look at what all values we have. So first is volume of distribution, which we denote as VT. It's given as 10 liters, and we know the clearance rate, which is 1 liter per hour. And we have to figure out what would be the half-life of this drug. So we already know this formula. T half, that is half life, is related to VD and clearance from this equation. T half is nothing but 0.693 into your volume of distribution divided by your clearance. You just put all the values in it and you will get the answer as 6.93 hours. Pretty straightforward numerical. If it's not clear, let me know. And I would anyways be sharing this PPT with you, so you can go over it again if anybody has not understood it right now. So for people who have doubts in these questions, we can clear it up right now, or in case you go through this PPT again and you come up with some doubts, we can also always take it up in the next sessions as well. I think this is the last question that I have for you today. In the upcoming assignments, we'll also be discussing some numericals which would actually help you in understanding the concepts of drug delivery a little better. So that's all the questions that I have for you right now. If people have any more doubts, you can let me know. Otherwise, I'll take up the second question. Somebody had a doubt here. Yeah. So the doubt is regarding the second question for bioavailability. The question is, we're giving 100 mg of drug and having 0.7 bioavailability, whereas for 200 mg having only 0.4 bioavailability. And how is this possible? Okay, I think you have the doubt is not exactly regarding the question. I'll just go over it. Okay, so you're saying, I hope I understand you correctly. You're saying 100 mg drug is given, the bioavailability is 0.7, and for 200 mg it is only 0.4. So your bioavailability will always depend upon what route of administration you're going through. So maybe both of these options, the route of administration could always be different. So if you have a different route of administration based on how you're delivering the drug, it will always have different bioavailability. So the drug that is, let's say, you give it orally versus you give it intramuscularly, they will always have different bioavailabilities. That's how the body is designed, right? So if you give a drug orally, it is going to go through some different path in the body, and then it will reach the site of action. Versus if you're giving a drug intramuscularly, that will be going through a different route of that will be taking a different path in the body, and that's why you never know how the drug is so the drug being lost will be different for an orally given drug and will be different for an intramuscularly given drug. So that's why you can have different bioavailabilities depending upon the route of administration. Is this clear? I hope I understood your problem first of all correctly. If Murari is there in the meeting, you can answer. Did you understand this, or should I go over it once again? If you're still in the meeting, you can let me know if that's okay. Okay, you understood. Okay, thanks. Someone is asking, could you discuss the third question again? Okay, let's look at the third question. Third question is the order of reaction. Okay, so for others, if you feel that this is a repetition, you can log off the meeting. We'll meet again next Tuesday at 6 o'clock. If you want to go over these questions again, you can stay back in the meeting and we can discuss more. So somebody is asking for this particular question. So how do we, which of the following equations depict a second order reaction? So first of all, the way you calculate an order of a reaction is you sum up the exponents of the concentration species. So concentration species are always written like this in square brackets. It means that the rate of the reaction is going to be dependent on a compound C1 and C2. Now how it is dependent depends on the exponents of these two species. So let's take first option. R is constant. It means that the rate of reaction will be constant for this particular reaction. It will not depend on any concentration of the particular reactant. Second option is the rate of reaction depends on C1 and C2. You take the sum of these exponents, it's 1 plus 1, it's 2. So this is a second order reaction. So this is how you calculate order of a reaction. It just tells you how the rate of reaction will be influenced based on the concentration of the reactants. Then you have option C, where you have R equal to K. C1 exponent is 1, C2 exponent is 0.5, C3 exponent is 0.5. You take a sum of these exponents, it's 1 plus 0.5 plus 0.5, it will again give you 2. So this is also an example of a second order reaction. So that's why the correct answer for this question is option B and C. Is this clear? Lisha, I hope I'm pronouncing it correct. This was your question, you wanted to discuss this again. So is this clear, or should I go over the question again? If anybody else has a doubt in any of these questions, we can go over these again. Or if anybody has missed any of these questions, if you have joined a little late and I was already discussing the other questions, you can also let me know. If anybody has missed the previous questions, I can go over them again. If not, we can stop the meeting. Or if you people have to discuss anything other than the questions, if you have any doubts regarding some of the concepts that we talked about or those were discussed in the first week, we can also talk about that. People who are joining right now, this might be a little late for you. I've already finished the session. You can join the other session, which is going to be from 7 to 9. Or if you want me to discuss some of these concepts again, I can do that as well. Thank you for the session. Okay, Lisha, I got your message. Okay. Thanks a lot for joining, and I can meet you all next Tuesday. Also, let me know if you want to go over some of the concepts which might be a little difficult in this session or in the next session. We can also discuss that. Let me know in the chat box. Okay, thank you. I'll be uploading both the PPT and the recording of this. I think you people will have access to both the recording as well as the PowerPoint presentation. Thank you for joining. I'll wait for a few more minutes if you people have any doubts. How can we get the PPT directly to our mails or any other way? I think yeah, somebody's asking how do we get the PPT. I think NPTEL people would have given you access to the Google Drive. That's where I'll be uploading all of these presentations. So you'll get it there. If not, then you let me know. I'll also share my email ID in case you people have any doubt. You can write to me, and in case you don't receive the presentations, you can mail me on this email ID. Yeah, you'll be getting the Google Drive link. I think you can access the presentations from that. Thank you. Any other doubts? If not, you can log off from the meeting and we can meet next week. Thank you. Those who are still in the meeting, do you have anything to discuss? Thank you all. Thank you for joining. Guys, do you have doubts? Or we can end the meeting. If you want, you can join the session at 7 o'clock and try that out as well. Okay, sure. I'll end the meeting. Thank you. I'll see you again next Tuesday. We'll be discussing concepts from week 2, as well as we'll discuss some of the new questions and hopefully some numericals as well. Thank you all. Thank you for joining.